What is phagocytosis? Think of it as the cellular version of eating. Phagocytosis is the cellular process of engulfing large solid particles, like invading pathogens and cell debris. It's a key function of our innate immune cells, such as macrophages and neutrophils. In the main illustration, you can see a large macrophage reaching out to trap and swallow a bacterium. The name itself gives away its job. It's derived from the Greek words phagein, meaning to eat, and kytos. Once the target is trapped inside a phagosome, it fuses with a lysosome, leading to the destruction of the invader by powerful enzymes. It is literally our cells eating away threats to keep us healthy. 1. Chemotaxis. Chemotaxis, known as the biological chase. Phagocytosis begins with a distress signal. Damaged tissue or invading pathogens release chemical signals. Phagocytes, like macrophages and neutrophils, pick up this scent. They migrate along the concentration gradient toward the highest source, often described as a biological game of hot and cold. Step 2. Adherence and recognition. Phagocytosis, the handshake. Once the phagocyte catches up, it must identify the target as an enemy. Receptors on the phagocyte surface bind to specific molecules on the pathogen. Opsonins, like complement and antibodies, coat the pathogen, creating a recognizable target marker. Opsonins enhance and boost the recognition and binding process. This initial physical connection is the crucial adherence step. Step 3. Ingestion and phagosome formation. Phagocytosis, the trap. Ingestion. Ingestion begins when the phagocyte membrane extends around the opsonized target. Pseudopodia completely surround the pathogen, creating an internalization. Once engulfed, the membrane seals, trapping the invader inside a membrane-bound bubble. This internal bubble with the trapped pathogen is the crucial phagosome. Phagosome formation marks the point where the pathogen is inside the cell. Step 4. Phagolysosome fusion and digestion. Phagocytosis, the digestive fusion, destruction. Lysosomes, containing powerful hydrolytic enzymes and acids, migrate toward the phagosome. The phagosome membrane fuses with the lysosome membrane, combining their contents. This newly fused vesicle is the crucial phagolysosome. Phagolysosome formation marks the initiation of pathogen destruction and breakdown. The combination of acidic conditions and enzymes begins to degrade the trapped pathogen. Step 5. Killing and digestion, oxidative burst. Phagocytosis, the digestive fusion, destruction. Step 5 is the actual killing phase within the mature phagolysosome. A key mechanism is the oxidative burst, a sudden, massive release of reactive oxygen species, or ROS. These ROS, like superoxide and hydrogen peroxide, are highly toxic and cause severe oxidative damage. They break down bacterial cell walls, proteins, and DNA, leading to rapid microbial death. This process, combined with enzymatic digestion, ensures total pathogen destruction. Step 6. Waste disposal, exocytosis. Phagocytosis, the cleanup phase. Step 6 is the final stage, involving the expulsion of undigested material. Once digestion is complete, the remaining inert fragments are sequestered in a residual body. This residual body moves to the cell membrane and fuses with it, in a process called exocytosis. The undigested waste is released into the extracellular environment. Exocytosis ensures the cell is clear of debris, restoring its environment and preparing for new targets. To summarize, in the phagocytosis process, attachment occurs through specific cell receptors bind the pathogen, for example, C3b. Ingestion. The phagocyte extends pseudopods, surrounding and engulfing the pathogen to form a phagosome. Phagolysosome formation. The phagosome fuses with an enzyme-filled lysosome. Digestion. Lysosomal enzymes and reactive oxygen species, or ROS, destroy the pathogen inside the phagolysosome. Residual body formation. Harmless undigested fragments are sequestered into a residual body. Exocytosis. The residual body fuses with the cell membrane and releases wastes, clearing the cell for new targets. Phagocytosis is also known as key immune defense. Specific cell receptors bind to pathogens, significantly enhanced by opsonization, which is coating with C3b and antibodies. Pathogens are internalized in a membrane-bound bubble, forming the phagosome. Phagolysosome formation. The phagosome merges with an enzyme-filled lysosome, bringing powerful enzymes and reactive oxygen species, or ROS. Killing and digestion. Lysosomal enzymes and ROS rapidly destroy and break down the pathogen within the phagolysosome. Exocytosis. Undigested fragments are sequestered into a residual body, which fuses with the cell membrane to expel waste, ensuring the cell is clear for subsequent targets. Critical for innate immunity, infection control, and presentation of antigens to the adaptive immune system.